you who are familiar, I guess you're in the geek squad, you know that SciCafe is, of course, some entertaining science. It's a lot of these fun drinks. And then hopefully, if I'm good enough, there'll be some questions back and forth. But for those of you who concentrate mostly on this part of the event, so enjoy yourselves. But tomorrow, you might spend a lot of time here. And if that happens, and you've got a killer headache when you wake up, you're going to reach for your medical cabinet and try to look for one of these magic pills to help you quench the pain of your hangover, right? You probably don't think about where these pills came from. And so these pills usually come from things that look like this. So most of the drugs that are in your medicine cabinet or at Duane Reade or Walgreens or that you get a prescription from your doctor, they come from natural organisms who are expressing different kind of compounds that we've sort of extracted these compounds and then figured out what they do that's helpful and beneficial to us. And so snakes, for the most part, have given us a lot of things that work on hypertension. And then an old favorite is the yew tree, which has given us, Taxol, which is being used in cancer. And then more recently, sea anemones are also giving us a lot of compounds and peptides for a range of different elements and ailments. These natural compounds are found all over the world, which is fun for us as scientists because we get to go out and explore them. But they've also found in almost all civilizations, and they go back for millions of years, even beyond recorded time. So one of the oldest texts that's found was from 2600 BC in Mesopotamia, and these are the clay tablets from those texts. And these clay tablets describe over 1,000 plants and plant extracts, such as poppy seed oil and cedar oil, things that we use today, even still. And so going through time, the Greeks, of course, the Greeks did a lot. And so here's Hippocrates of Cos. So he was the drug guy for what was going on in Greece, pushing lemon juice as a laxative, and he was pushing olive oil for healing wounds, right? In China, we have the Wu Shi Er Bing Fang, which thankfully translates to 52 diseases. I wonder how they got that nice round number. I was like, 52, not 53, not 51. In this text, they likewise describe a whole bunch of plants and organisms from which they were able to get extracts that were used for medical benefits. And so even though all of these civilizations were independently evolved, the medical treatment that they've used seem to have converged, and they use very similar things, actually. So Hippocrates was using this white plant for inducing vomit, and at the same time, the Chinese was using the black version of this plant to induce vomit. And so that shows you that even though we're all from very different cultures, we've somehow migrated to nature, and in nature we found different compounds that are very useful for treating several different kinds of ailments. And this use of using natural products, of course, continues today. And so these are all organisms for which you can either get commercially available products or have products that are in development. So the Gila monster is being used for diabetes treatment. Chinese red-headed centipede, they've found new work that looks like it's doing interesting things for pain. The poison dart frog also is doing really nice pain research currently. And the Brazilian land snake has been doing hypertension. While these animals are great, and certainly most of them are venomous, there's one poisonous one. And so most venomous organisms, when you think of them, you're thinking of snakes or spiders or scorpions. Not many people are thinking of snails. And when they do think of snails, you're usually thinking of this garden variety sort of land snail. So those, or if you're a foodie, anybody in the audience, you're thinking about this version of snail, the escargot, right? And so my snails are not those. They've gone bad. They're tattooed with leather jackets. These are the Brooklyn hipster snails. I'm from Brooklyn, and so it's a good thing. And so these snails are sort of on a feeding frenzy, and this is what they're doing. So hidden under the sand here, it's a snail. The orange thing going out is like a tongue. It's called a proboscis. On the tip of the tongue, it has a dart, which is filled with venom, and it's going to inject the venom into that fish. And this is real time. We haven't sped it up. Yeah, isn't that awesome? I love that. So when I saw that, I was like, I have to work on these guys. So it's going to swallow the fish completely. Awesome. That's what I wanted to hear. Completely swallow the fish and have itself like this ridiculously impossible meal that no one would expect it to have, right? So you're going to see it again. So they can usually swallow a fish about their body size. So this is Conus bullatus. We've dug him up from the sand. And again, that little proboscis injects the venom. So what's happened to the fish? Paralyzed, right? It's gone completely stiff. You've never seen such a stiff fish in your life. It's completely stiff, but it's not dead. So the snails, when they inject the venom into their prey, they're basically working to shut down the function of the prey, but they don't kill them necessarily. So if you focus near the throat area, you'll see the fish will try to recover. It's too late, not getting out of that one. It's like that blind date that you can't get away from. You're like, oh, I'm committed. And so what's happening in the venom is, I like to say that it's sort of like a cluster bomb, and this is like a man-made cluster bomb, but these venom from these animals are natural cluster bombs. And so the way that a cluster bomb works is that, you know, one big bomb gets explode, and then the other little bombs come out, and they just cause havoc everywhere. So instead of having these little tiny vesicles of terror, what the snails and other venomous organisms have in their venom are all these little clusters of peptides. So the venom is actually a cocktail, similar to these. So the cocktail is made up of not just one single compound, but it's made up anywhere between 50 to 200 different compounds together. And in our group, we mostly focus on peptides, so that's what I'll talk about, but there are other kinds of compounds in the venom as well. And I should warn you that they also are fatal to humans. So they don't just feed on fish. They are also fatal to us. They don't swallow us, of course. We're a little too big, but they can stop us from breathing and our hearts from beating. All the things we need to live, right? And so the way they do that is by invading the cells and basically trying to turn on and off the way cells function. And so by doing this, they inhibit the natural physiology of cells. So if I injected all of the cone snail venom into you, it would kill you. But so we don't do that. So what we do is try to identify the individual peptides in that cluster bomb and see if they can work as a switch to turn on and off diseases. Because if you think of a disease, such as pain or cancer, they're really cellular switches that have gone wrong. So in pain, for patients who are experiencing chronic pain, there's a signal going from that pain area to your brain all the time saying, I'm in pain, I'm in pain, I'm in pain. And your brain tries to shut it off, but it can't, because for some reason that reverse signal to wherever the pain is doesn't get processed. Similarly with cancer, so a cancer cell is basically like your cells on crack. They're going crazy. The signal is broken, and so several cells are starting to multiply, and then tumors divide, and then they spread from one area to another area. And so in a very simplified version, this is basically on-off switching. So it's on-off switching of how cells function. And what we're trying to do with the peptides in the venom is to find the lever that will control those switches. And so over the years, we found that venom peptides are very good levers for controlling this on-off signal. I learned all about venom work from the guy I call sort of the godfather of snails, and that's Toto Oliveira. In the 80s, Toto extracted some peptide from Conus magus, and this became the first drug that is commercially available on the market from cone snails that you can use, and it's used to treat chronic pain in HIV and cancer patients. And so Prealt was fabulous, right? So it was sort of like a breakthrough drug, because prior to Prealt, most biopharmaceutical companies, when they were looking for things that alleviated pain, they were looking at small molecules, and specifically they were looking at things like morphine and things that worked on opioid receptors, right? So no one was looking, certainly not at peptides, and certainly not at things that did not work on opioid receptors. So when Prealt came out, it was definitely a breakthrough, because not only was it giving us new compounds, it was also giving us new targets, which means that we can, solve the problem in many more ways than just hitting the same nail on the same head over and over and over again. But before we could break out the champagne and celebrate, there was a problem. Don't run out and buy Prealt stock, not yet. So it's 25 amino acids. This is the chemical structure. I am a chemist, so you had to see a molecule tonight. So this is the chemical structure of Prealt. It's made up of 6 cysteines that are connected. And the major issue is, because it's so complex, it doesn't cross the blood-brain barrier, so you have to take a spinal tap to get it. So that's why I said don't run out and buy stock, right? So unlike a morphine drip is very easy because it works in the blood system and you can hit the button and dial up for as much as you want. You can't do that with Prealt. And so it's kind of like trying to get a camel through the eye of a needle. And so in the lab, what we're doing is this sort of Trojan horse strategy. We're trying to trick the Prealt across the blood-brain barrier so that we can get it into the system without having to give people spinal taps. I'm calling this Building a Better Mousetrap. This is the name we used on our NIH grant. So MOUSE stands for Marine Organisms Used in Shuttle Encapsulation. So in this research, what we're doing is we take the peptides, and they're basically being developed inside of nanoparticles. We have the peptides sort of growing recombinantly, so it's a peptide factory on the inside. Once we have the peptides on the inside, we add funky groups to them, to the particle, which then shuttles it across the blood-brain barrier. And then once it's across the blood-brain barrier, we then use chemical technique to sort of break apart the nanoparticle and release the drug. And so we've tried this. We proposed it, and then we actually tried it, and it seems to be working. And so if we're able to do this on rat brains, the next step, of course, is to try it on human brain cells, and then also to see if when we trigger the release of the drug, that it's still active, right? Because the worst thing is if you do all this and the drug doesn't work. But that's called science. So we've done this part, and so there's still a lot more to go, and we're really, really excited. But this is our first endeavor in how we can take what we're getting from the venom peptide and actually make it into an actual therapeutic drug that will help people in a beneficial way. Recently, not only did we find that a lot of the peptides in snails were neuropeptides, and so they were turning off and on neuronal signals, but recently he found some insulin-like molecules in cone snails. So this is Conus geographus, and so this snail is what we call the cigarette cone, because essentially you have about 2 hours to smoke a cigarette before it kills you, if you should ever get envenomated by these cones. And so the cone snail insulin seems to be doing the same thing. It's regulating to bring your blood sugar down. So this is awesome, because now not only are we able to turn off switches on things that are neuronally driven, we can also find out that we can turn off switches on things that are metabolically driven as well, right? So the Venom is sort of turning out to be a great case study or a great model system, not just for the things that we thought directly related to it, which are neuropeptides, but things that are also seem to be more universal to cellular function as well. So this is great. So hopefully, in another 24 years, there'll be a nice, perfect Conus geographus test for lowering blood sugar. And so mostly I've been talking about cone snails, but these snails come in 3 different flavors. So cone snails, because they're shaped like an ice cream cone, right? But you don't want to lick these. And then there's terebrids. Those are the group that my lab personally works on. And then we have turids. So there are 3 different physiologies for terebrids. And the ones that we're interested in are the ones that are most similar to cone snails. And so therefore, they're the ones that have the same venom apparatus. And they're the ones that we know for sure are producing peptides to subdue their prey. So the way that traditionally the work used to be done is that you would go to the beach and collect as many of the snails as you can find. And you bring them home, crush them up, dissect out the glands, and you go to work in identifying what those components are. So that's the traditional way of doing venom discovery. And it was mostly driven by size and quantity, right? Because you needed a lot of them in order to do the chemical and analytical characterization that was necessary. What we've been doing is what I'm calling venomics. It's the X, you know, like genomics, transcriptomics, metabolics, everything. So in the venomics approach, we first start by letting nature tell us what it's doing, right? So we build a family tree of snails, and from this, we can then do more targeted hypothesis-driven research, and this then is driven by evolution and diversity. So for example, let's say on that second branch, one of those snails were the ones that were responsible for the Prealt drug, and we wanted to find other kinds of Prealt drugs, but ones that maybe could cross the blood-brain barrier, so you didn't have to take a spinal tap, then we would focus on looking at all the snails that are on the same branch as Prealt, right? So by first concentrating on what nature is doing and building this phylogenetic tree of terebrid snails, we now have a very smart roadmap for how we can go about and look at venom diversification throughout these animals, and also as we go along and identify the different components that are in these animals. And so in order to build the phylogenetic tree, you need samples, right? So that means field work. So this is the good part, and this is why I love being at the museum too, because we get to do expeditions. You go out all around the world and you're collecting things. Our most recent expedition was to Papua New Guinea. And so in Papua New Guinea, we boarded a plane. How do you get there? You board a plane and you sort of fly off to an island, and then once you get there, you board another plane and you fly off to an island off of that island, and then you get onto a truck and you drive through mud to the very tip of the second island, and you take a boat for about 2 hours till you get to the island off of the island off of the island. And so we are now here at that spinning red star, which Google can't even find. And so that's where we were camped. And so I figured out the secret to actually getting tenure and keeping your grad students happy is to go to that island off of the island in the middle of nowhere and drop them into the ocean. So we go out and we collect the snails, and then when we get them, we come back and we are doing this Venomics strategy. So just to describe a little bit more about what Venomics is, I like to call it a marriage. So it's a marriage between something old, something new, something borrowed, something blue. And so we'll start with the blue. The blue is usually the grad student who gets left home. They're the student who's not on the island, off of the island, off of the island. And so the new are all of these really great technologies that are happening, both in mass spectrometry and also in sequencing. So all these advances that are currently coming available are allowing us to explore all these different venoms that we previously could not. So you remember when I said the old strategy was driven by size and quantity? That was because prior to these genomic and proteomic techniques, we had to collect anywhere between 80 to 100 different specimens of the same species. With these techniques, we now collect 5. And that's mostly because grad students lose them. So we have 5 samples, 2 for replication, and then the other 3 because somebody will lose it. And so in this way, we're not only conserving the organisms that we have on our planet to make sure that not completely everything is going to go extinct from drug discovery, but we're also able to do very powerful techniques. We don't need as much, and we are getting a lot more information. And then so the old, of course, are all of these organisms who have been doing this for millions and millions and millions of years, right? And so what we're trying to do is figure out how they're doing it, how they're doing it so effectively, and can we replicate it in the lab? So the first part was building the phylogeny. So this we've done. And so this is the phylogeny for terebrid snails. So everywhere that you see black means that they have that venom apparatus, similar to cone snails. Everywhere that you see white, they don't have that venom apparatus. So just in building the tree, we've already been able to figure out which organisms are the ones that are producing peptides, that we know are actively producing peptides, and which ones are not. So then the next step is to do transcriptomics and the proteomic work, which is basically sequencing and mass spec. And so for the sequencing effort, what we do is we extract out that venom gland, right? And then you're basically breaking apart this jigsaw puzzle. You put it into a machine, it breaks apart the sequences. Messenger RNA is what we're using. And then you have this red and blue jigsaw puzzle of millions and millions of pieces that you then have to piece back together. So we call that assembly. So we assemble the jigsaw pieces of the puzzle back together, which is the genetic story of the snails. And we're taking it specifically from the venom gland, and we call that a transcriptome because it's going to transcribe only the genes that are being expressed in that tissue, right? And then once we have that transcriptome assembled, we then go through and do a bunch of series of different analyses. So we do the phylogenetic analyses, we identify the peptide toxins, we annotate what else is in the venom, because similar to snakes and scorpions and spiders, a lot of the venom components that we find in snails are also found in those other organisms. And that's because we've discovered that there's convergent evolution of venom within these organisms. And so if we get lucky, we'll end up with that little pill on the bottom, so that means that we've found the compound that can be used for drug discovery. And so this work is actually very hard and very complicated and takes a lot of time. And that's why it's called next generation sequencing. We'll need like next generations of grad students and postdocs to actually get success, but we're on the path and we're excited. And so we did the transcriptome analysis of these two species, Terebra subulata and Treptylostephanus annealis. We then compared it to cone snails to see if we're finding either the very same things as cone snails or that we're going to find something different. What we happily found is that we're finding very, very different things. These are the putative toxins that we found from our assembly, and then we blast them to conotoxins, and they also blast to those, but a whole bunch of them don't blast to conotoxins, and they blast uniquely to a terebrotoxin database that we have. So it's kind of like Batman versus Superman. They're both great, but they're working in different ways, and that's great news for us because it means we all can stay employed. And so because this assembly is from scratch, right, there are no references to compare it to, and snails are not model organisms. So when you're doing biomedical research, you use these things called model organisms, not these models, right? But these models. So you're always injecting into fruit flies or mice or C. elegans, and so there's a whole system if you want to use model organisms. If you don't want to use model organisms, you're sort of on your own, and you have to go out and find the tools that you need, and you have to also validate that what you're finding is correct. And so to do that validation, we then are combining our transcriptome assemblies, which are totally non-referenced, with proteomic work. And so this is work in which we do with mass spec. And in collaboration with a good friend, Trixie, down at NYU, is how we're going to validate that what we're actually finding is legit. So we've done this for one of the snails, another snail, Terebra variegata, in which we extract the venom, same from the glands, but instead of putting it into the sequencer and doing next generation sequencing, we're putting it into the mass spec, and we're actually characterizing all the things that are there. So unlike where the sequencing is maybe it happened or maybe it didn't, the mass spec tells you for sure it's there. So we do that using two different techniques, and so from that we identified this peptide for TV1. The peptide needs structure to function, so initially it's linear, and then we turn it, when we fold it, it becomes making the disulfides connect, is what we call fold it. Then it has the function. If it stays linear, it doesn't function at all. And so TV1 has three different possibilities for how it can fold. And so how do we figure out which fold is correct? Is a lot of chemistry. And so we did that. And to confirm it, you do something called NMR, which is very similar to the MRIs that you do at the hospital. And so we did an NMR, and we have now the solved structure for TV1, and it matched exactly what we found with the mass spec data. So this was really, really fun and great because we've gone from the transcriptome and sequencing the DNA, finding sequences, validating them in the mass spec, and then confirming what we have, both in structure and fold, and now we know that we have a really legit peptide from this snail. And then the biggest news, breaking news, we found out that this peptide is also active in cancer cells. So this is the first patent from the lab, and we're really, really excited because we've just shown here on your y-axis is the viability percentage, and on the x-axis are different concentrations of TV1 that we've tested in mouse models. So at different concentrations, you see that TV1 is killing cancer cells and not normal cells. So we're like, celebration! We're very happy. And so what we also were happy about is that unlike the drugs that are currently on the market, so that's all the way here, like doxorubicin, they don't discriminate between cancer cells and tumor cells. They kill everything in their path. And that's why for cancer patients, their treatment is always so horrible, right? Because their whole body is under attack. And so we were super excited because our peptide doesn't do that. It seems to be doing discrimination. It's not a great job, but it's better than what's currently on the market. And then what's also exciting us is the fact that this peptide is very specific to liver cancer. So in this plot, On the x-axis are all these different kinds of cancers we tested against. On the y-axis is the cell viability. And so where you see the brick hashtags, that means nothing, the cells haven't been treated. Where you see the polka dots, that's our peptide, TV1. And where you see like the gingham checkered thing, I can't even look at it, that's the doxorubicin. That's the stuff that's currently on the market. Our TV1 doesn't seem to do much with cervical cancer, neuroblastoma, or prostate, but it's doing something with liver. It's not doing as much as doxorubicin, but that now has given us a platform that we can now tweak. And so we can attenuate that on-off switch so we can get it to do as much as doxorubicin or even better. And so not only have we identified a peptide that is selectively killing cancer cells and not normal cells, it's also selected for just liver cancer. When you're making a drug, one of the things you want to do is to make it as specific as possible. So like when you're taking that drug tomorrow, if you've had too many cocktails on the Geek Squad, you want it to cure your headache. You don't want to make it, you don't want to have to run to the bathroom for diarrhea also, right? And so that's what being specific to liver cancer helps us to do. So to kind of summarize the terebrid story so far, we've used this evolutionary-driven discovery method to identify the lineages of peptides which are actively producing toxins from those that don't, and we've started to characterize the function of these toxins. But our snails aren't the only ones, right? So here I'll show you these. These are vampire snails. Love these. They're on your left. So it's sticking out its proboscis. It's going under the gill of the fish, and it's basically going to suck the blood of the fish, right? So on your right, you can see it's actively sucking the blood. So what kind of drugs might we be able to find from these snails? Anticoagulants, right? Exactly. Anticoagulants, blood thinners, all of these are great things to be exploring from these snails. And so similar to what we're doing with our terebrid snails, we have a collaborator in the University of Rome in Italy, and they've started to identify the different components, both using transcriptomics, and we're helping them with some of the proteomics stuff as well. And they've identified several compounds that seem to be, because why isn't the blood clotting, right? And so these snails don't kill the fish. They just sort of suck on the blood, you know, have a meal. It's a blood meal. And then they move on. And so it's an exciting area also, because we're trying to figure out what's going on here. And so I've mostly talked about snails, but there are all of these different organisms, and all of them seem to be doing a similar thing, which is basically invading their prey and figuring out how they can turn off switches so that they can have themselves a meal. And so snakes, scorpions, spiders, sea anemones, all of them are producing compounds. And so in general, these venom peptides turn out to be very, very good levers for controlling cell disorders. And so we're very excited about what's happening, and that's sort of how you get from mollusks to medicine. And I'll leave you with this last video. This is Conus tulipa. And so this guy, we call them the net hunters. So they basically will stick out their mouths really, really, really wide, and they will swallow like a school of fish and then individually harpoon them once they're inside of their mouths. It's like truly amazing stuff. So thank you, and I'll take questions.